Happy Saturday, Brain Bee participants. I hope you are having a great day thus far. And while I'm sad that I can't be there today to tell you about my research passion, the eye, well, specifically the photoreceptor cells, I'm still really excited to get to talk a bit about the eye and then show you what I get to do on a daily basis. Oh, and yes, this is me, so you now have a face of the person who is going to be speaking to you for the next 10 to 15 minutes. So let's jump right into it. So as we all know, the structures and functions of the eye are pretty complex. Each eye constantly adjusts the amount of light it lets in, focuses on objects near and far, and produces continuous images that are instantaneously transmitted and processed by an intricate system of interconnecting neurons, which, begins in the retina, which is in the eye, and ends with the visual processing center in our forebrain, which is the visual cortex. So what are the parts of the eye that must work together to produce clear vision? First off, light enters the eye through the cornea, this clear curved layer in the front. The cornea serves as a protective covering for the front of the eye, and it also helps focus light on the retina at the back of the eye. After passing through the cornea, light must travel through the pupil, this black dot right here, and the iris, this circular colored area of the eye, which controls the amount of light that enters the eye. The iris allows more light into the eye, enlarging or dilating the pupil when the environment is dark, and allows less light into the eye, thus shrinking or constricting the pupil when the environment is bright. So do you know which muscles control the size of the pupil? Give you a couple seconds to jog your memory. So I'm hoping you got it, but it's the iris sphincter and the dilator muscles. Extra points, I won't tell you the answer, but do you know what the substance is that fills the anterior chamber between the lens and the cornea? And what is that called, and what is its job? If you don't, maybe just check it out later if you're so inclined. Behind the iris sits the lens. By changing its shape, the lens focuses light onto the retina. Through the action of small muscles, called the ciliary muscles, the lens becomes thicker to focus on nearby objects and thinner to focus on distant objects. Also, behind the lens, there is this clear jelly-like substance called the vitreous, which fills up the space behind the lens and holds the eye in its spherical shape. Next, we're going to look at the retina, the light-sensitive, multi-layered tissue that contains the light-sensitive cells, photoreceptor cells, and the blood vessels known as the choroid that nourish them. The most sensitive part of the retina, as you may know, is a small area called the macula, which has millions of tightly packed photoreceptor cells. And do you know what type of photoreceptors are found within the macula? Yes, you do. It is, of course, cones. We have 3 flavors of cones. We have blue, red, and green. And the high density of cones in the macula makes colored vision very detailed. Besides cones, we also have rods, which are responsible for night and peripheral vision. Rods are more numerous than cones and much more sensitive to light, but they really do not register color or contribute to detailed central vision as cones do, which we just described. The RPE, located between rods and cones, and the blood supply of the choroid also perform really critical functions that support rod and cone health and integrity. Can you think of any of these functions off the top of your head? Off the top of my head, I can think of light absorption and phagocytosis of the outer segments of the photoreceptor cells. In the subsequent layers of the retina, we have bipolar cells, and these cells take the electrical information from the photoreceptor cells and pass it along to other retinal cells. And these bipolar cells make connections with downstream retinal ganglion cells. And these cells then extend to form the optic nerve, which conveys visual information such as shapes, contrast, and color to the brain. There are 2 other retinal cells I did not mention. Can you name them? The other cells are horizontal and amacrine cells. However, I forgot to mention one other cell, so like extra points if you can think of this one. Yes, it is the retinal glial cell. The Mueller glial cell is the last cell of the retina. So with that, congratulations! We have come to the end of my background section. So while the eye is pretty fun to talk about because it's one of the main ways we interact with the world, as a neuroscientist and retinal researcher, I really do enjoy getting to explore the eye up close and personal. So my next round of business is to kind of show you my lab space and kind of how to dissect a retina. Welcome to our vision lab. Come follow me to see some of what we do. On the right, we have our -20 and -80 degree freezers, and on the left, we have our 4-degree fridges. This is where we keep most of our retinal samples and solutions. So, while I get prepared for our eye dissection, I just wanted to show you a little preview of what a university lab looks like. Done! I'm ready to dissect. Let's head over to the dissecting station now. And this will be the area where we will be isolating the eye and dissecting the retina. We typically perform retinal experiments on both pigmented and albino mice. So here I thought I would show you me doing the nucleation process on both types of mice. And in order to nucleate the eye, you first have to push the eye out of the socket with your hands. And then with curved scissors, you can carefully cut the optic nerve under the eye. Next, I'll transfer the isolated eye into a petri dish, and since I'm isolating the retina, I'll take care to only touch the cornea or the sclera, that is the outside covering of the eye. So here, you can clearly see the transparent cornea, the iris, which is brown, as well as the pupil. In order to isolate the retina, I first need to remove the cornea and the iris. To do this, I must make an incision at the corneal sclera junction and then cut in a circular motion around that barrier. So here you can see that I've isolated the dome-shaped cornea as well as the iris. Next, I must remove the lens and the vitreous. So as I am using my forceps to pull the lens away from the retinal cup, I will also be pulling off shreds of the vitreous, which you can see to the left of the isolated cornea and iris. And now I have just placed the crystalline lens right next to the vitreous. Something you might not have noticed is that the mouse lens is closer to spherical in shape than the human lens, and it occupies most of the vitreal cavity. This arrangement in rodents is reminiscent of the fish eye and should provide uniform optical focus over a wider field of view, which would be advantageous to a small prey animal. And here you can see the retinal pigmented epithelium. It is the dark blackish blue pigment that lies right beneath the sclera, which is a clear protective layer surrounding the eye. Once I have the eye cup ready and the lens and vitreous are removed, I next will isolate the retina. So what I do is this peeling method where I grab the sclera and the RPE and I carefully peel it away from the retina, and this is the final product. Since enucleating the eye means cutting the optic nerve, I wanted to make sure that you got the opportunity to see what the optic nerve actually looks like. So here again, we can see the cornea, the iris, the pupil, and the RPE under the sclera. But now we can see the optic nerve as well, which, consists of all the retinal ganglion cell axons. So in this lab, we use multiple techniques to study the retina. One technique that we use often after retinal dissection is cryosectioning. So once we create an eye cup like the one I showed you previously, we end up embedding the eye cup in a solution called OTC compound, and then we end up freezing it in liquid nitrogen. After freezing, we section the retina on a cryostat, and then once the retina is sectioned and mounted on a slide, we can either stain it to identify certain proteins, or we can just look at the architecture of the retina under normal light. And that's what I'm going to show you in a moment. Hi, so here I am at our trusty microscope, and I am going to look at some frozen retinal sections and share them with you. So this is a depiction of what the retinal sections look like at very low magnification. And with greater magnification, you can see all the layers of the retina, starting with the retinal pigmented epithelium, followed by the photoreceptor cells, and then the bipolar cells, and last but not least, the retinal ganglion cells. Cool, right? While I really enjoy studying the retina and think it's, pretty fun to do research, one of the coolest things we get to do in our lab is to dissect retinas in the dark. So the reason why we do experiments in the dark is to also understand the basal dark state of rods and cones. So in order to do that, we've built a dark room and we use night vision goggles and infrared light to see what we're doing. Thank you so much for coming to my station to learn a bit more about the eye, and I hope you had as much fun as I did. Have a wonderful rest of your afternoon, and please do not hesitate to reach out to me at my email, kcvrow@usc.edu if you have any questions.